Hello friends, welcome to this session. In today's session, we are going to learn about steroids. The learning objective is basically nomenclature, stereochemistry, and metabolism of the steroidal sex hormones. Steroid comprises group of cyclical organic compounds whose basis is the characteristic arrangement of 17 carbon atoms in a 4-ring structures linked together from 3 6 carbon rings and a 5 carbon ring. An 18 carbon side chain on carbon 17. So this is the backbone of the steroids. This is a cyclopentane per hydrophenandrin backbone. The steroid hormone generally combines with the steroid receptor and the complex binds to the receptor, enters to the nucleus and combines with the sites on the chromatin, activating mRNA transcriptions. Classification generally these are classified as the sterols, sex hormones, cardiac glycosides, bile acids, and sapogenins. Out of these, sex hormones is important from syllabus point of view. Sex hormones are usually classified as the three classes: androgens, which are also known as the male hormones, androsten and testosterone are the examples of this class. Estrogens, these are also known as the female hormones: estrone, estradiol, stilbesterols, exesterol. These are the examples of female hormones. And gestrogens, these are the corpus luteum hormones. Example is the progesterone. Nomenclature of steroid designated by the tribal names. Discard the names in favor of the systemic names which are approved by the IPAC. The nucleus and numberings, there are 4 rings which are fused together and that will give the backbone which is known as a cyclopentano per hydrophenandrins. These are to be categorized as ring A, B, C and D and other side chain at the carbon 17. The beta configuration, whenever there is a substituent attached on the ring systems and which is above the plane, then it is designated as a beta carbon or in the dark or thickened line. Alpha configuration, generally those substituents which are attached in the ring system from below the plane of these rings, designated as alpha or broken or dotted lines. While unknown configuration are shown like this as a size, and one should specify the chemical configuration of the sterols. If some of the carbon atoms are missing in the steroid, the nomenclature of the remainder will not change in order to illustrate the above rules. Generally, the nomenclature remains same. Just we have to the numbering remains same. Just we have to use the prefix. Suppose angular methyl group is absent, and also numbering won't change. That will be just dominated by the NORs. If the side chain does not contain a methylene group, this is indicated by the prefix nor preceded by the number of carbon atom that has disappeared. So this is 21st carbon. If it is missing, it is to be known as the 21 nor. If the steroid does not contain angular methyl group, this is again indicated by the prefix nor. If contraction of the ring of steroid, this is again indicated by the prefix nor preceded by the small capital letter indicating the ring affected. It is here the ring A is contracted and it is mentioned as A nor. If enlargement, then it is the homo preceded by the capital letters indicating the ring affected. So here ring B is enlarged, so it is shown as beta homo 5 beta pregnant. If ring fission in steroids with the addition of the hydrogen atoms to each new terminal group, this is indicated by the number showing the position of the bond broken. So here position 2 3 there is a breakage of the bond, so it is a 2 3 seco 5 beta androsten 2 3 dioic acids. If the steroid contains 3-membered ring, this is indicated by the prefix cyclo preceded by the member of position concerned. Due to the introduction of the above mentioned rules of nomenclature, some names are employed in the earlier literature have been rejected and replaced. For example, caprostane is now called as a 5-beta cholesteins. Also, compounds are now called as the cyclocompounds. Now, the methyl groups are normally present at the carbon 10, carbon 13, and alkyl side chain at carbon 17, while the sterols which at carbon 3. If one methyl group is attached to the carbon 25, it is assigned the number 26. If there are both are substituted, then carrying the substituent sites first in the alphabetical orders. is assign the lowest numbers. So if look at here, there is one methyl and ethyl. So alphabetically E come first, therefore the 26 number is given to the ethyl carbon. When the configuration is not known for one or more center, it is indicated by the Greek letter epsilon, prefixed by the appropriate locants. If one of the breach hydrogen atom is replaced, it may be desirable to restate the stereochemistry. If 9 alpha fluoro, 5 alpha pregnant instead of 9 fluoro, 5 alpha pregnant. So whatever the hydrogen is replaced by fluoro, it is indicated whether it is alpha or beta. This is 5 alpha choline, 5 alpha cholesterol. Unsaturation indicated by the changing the N to the in. If it contain one double bond, it is contain in. If it is two double bond, then it is dine and so on trend. If there is three double bonds, the delta symbol is used to designate the carbon-carbon double bonds. So carbon-carbon double bond between the 4 and 5 is referred as delta 4, while between 5 and 10 is designated as a delta 5. If the double bond is not between sequentially numbered carbon, both the carbons are indicated in the names. When an additional ring is formed by means of direct line between any two carbon atoms, steroids or attached side chain, the name of the steroid is prefixed by the cyclo. This is prefixed by the number of positions of the lines given by the new bond. Greek letter alpha, beta, epsilon denotes the configuration associated with the bonds. Naturally occurring steroid contain one or more additional heterocyclic ring fused or attached to the D formed by the modification of the side chains. For example, cardinaloid that contain lactone 5-membered or biphenoloid it contain pyranone 6-membered ring systems. Lengthening of the side chain by insertion of the CH2, one or more CH2 group indicated by the prefix homo. Removal of terminal ring with the addition of the hydrogen atom at junction where the adjacent ring is indicated by the prefix des, followed by the italic letter designated the ring. So here ring A is completely absent, so it is to be denoted by the des A. Alternative application nor homo is the above systems. The systemic names are based on the stem name, which in turn based on the hydrocarbon system. So 27 carbon is known as cholestane, 21 carbon pregnane, while 19 carbon androstane, 18 carbon estane, and 17 carbon as a gonane. Now the stereochemistry of the rings markedly affect biological activity of the given class of steroids. There are 6 asymmetric carbon atoms, 5, 8, 9, 10, 13, and 14. The nucleus therefore gives 64 optically active forms. It can be exist in two conformations, namely chair and boat conformations. Chair conformation more stable than the boat conformation due to the less angle strains. Hence, all cyclohexane rings exist in the chair form. Hydrogen or functional groups on the beta side of the molecule are denoted by the solid lines and the alpha side denoted by the dotted lines. It can exist into two conformations, namely chair and bed forms. Hydrogen are functional groups on the beta side denoted by the solid lines, while the alpha denoted by the dotted lines. AB trans ring A and B, these are transform the hydrogen atom at carbon 5, that is this hydrogen has a alpha configuration while the carbon 19 having beta configurations, and therefore it forms trans to the ring junction A and B. The ring B, C and CD, these are the trans, the configuration of 8B, 8 beta and 9 alpha hydrogens and 14 alpha hydrogens and carbon 18 angular methyl group denotes this transfusion for the ring BC and CD respectively. Cis and trans relationship of one or four rings may be expressed in terms of the backbones. 5 alpha cholesterol have a trans anti-trans backbone, while the anti denotes orientation of the ring that are connected to the each other and have trans type of relationships. 5 beta cholesterol has a cis-syn trans anti-trans backbone in which the AB rings junction fuse is cis. The term syn is used in the similar fashion as anti to define the cis type of relationships. So there are 6 asymmetric carbons. There are in the cholesterol molecule there is total 8 configuration, 8 asymmetric chiral carbons, and therefore it will give the total 256 optical isomers. If the chiral center in the side chain contains carbon 20 is also included, the number of possible optical isomer will be 512. Two type deals with the fuse ring configuration of the substituent at carbon 3 and carbon 17. In the fully saturated nucleus, there are 6 chiral centers and therefore 64 number of optical isomers are thoroughly possible. It is worth mentioning that only few exist actually, while many cannot exist due to the steric limitation. Oxidative degradation methods gives a great deal of evidence for the stereochemistry of the nucleus. In AB fusion, both cis and trans were found in natural steroids. X-ray analysis reveals that the fusion of the ring B and C was trans, while steroid is a plate which can be explained only if the fusion ring B and C has occurred together in trans manner. Only trans BC fusion takes place in a natural steroid. Ring CD possesses transfusion. Mostly steroids have the trans CD fusion, exception in cardiac glycosides and toad poison, in which there is a cis hydrogen at carbon 9 is a trans to methyl group at carbon 10. The methyl groups at carbon 10 and carbon 13 are cis. NMR spectroscopy is quite useful to steroid chemistry. Angular methyl group undergoes the coupling with the certain protons on the steroid nucleus. It is also found that peak width of the angular methyl group half height for the transfusion isomer is larger than that for the cis-fused isomers. So if it was trans and cis comparison, so cis having half the angular peak. Configuration of hydroxyl group at carbon 3. Beta series found in all the natural steroids. The prefix beta indicates that it lies above the plane of the molecule. If the hydroxyl group lies below the plane, it gives the rise to the alpha series. So thank you very much. In the next session, we will see the SAR and metabolism of the steroid hormones. Thank you.